Hi guys, today we're going to be working on a DNA profiling lab. You should have already read the background information, taken a look at the setup of the lab, and answering your pre-lab questions should already be done. So right now I'm just going to show you kind of the procedure, how you would go through this if you were doing it yourself, and then we'll take a look at the results after that. Some background information on what I've already done is I have already made these agarose gel beds. So this is made out of something called agarose, and it also has a material in it called CyberSafe. And the CyberSafe is something special that makes it so that when we actually run our gel with the DNA samples, it will make it so that we can see the different samples, the bands, and we'll talk about that in a minute, under UV light. And so it makes it so that we don't actually have to wait a really long time to see our results. We can kind of see them right away, which is nice. So I'm going to kind of walk you through the setup. I'll show you how we load our samples into our bed. We'll turn it on, and then I will show you what the results look like after that. So our first step is I've made these gel beds, and they're sitting in their molds, and I need to get them ready to load. And so to do that, I have to take off these two rubber bumpers on the end. I want to be really careful because if I just kind of rip them off, my gel might split and rip. So I can do it gently, but it comes off pretty easily. I'm going to take both of these off. Otherwise, it won't fit into our little box here. So we just need to take that off. And then this blue thing here is called the comb, and it actually makes the little wells inside the gel. So it goes into the gel just a tiny little bit to create a well that I can then put my liquid samples into. So again, I'm going to be careful taking these out because I don't want to rip the wells at all. All right. Now that I have that out, I'm ready to put this into the box that will eventually run my samples. This little plastic piece actually stays on and it fits right into it so that it doesn't move around too much. So I'm going to put it right there. The notch makes it really easy so that our sample runs in the correct direction. But what I'd like to point out to you is there is a black electrode on this side and a red electrode on this side. And the red one is where the positive electricity is going to be, and the black represents the negative. And this is important because DNA is negatively charged, which means when I turn this on, it's going to be attracted to that positive electrode. And this is good because this means my samples will run the correct direction, where I have a lot of space for my samples to move. If it ran backwards, my samples would pretty much run right off the gel almost immediately. So it's really important that you get it set up correctly. This is a little bit foolproof because it has the notch in it, but it's definitely possible to mess it up and your sample would run backwards and you would end up with nothing on your gel. So now that I have this in here, I'm going to put my buffer in. So this buffer is also what is in the gel. And this needs to get put into the gel box in order to do this lab. So you need to just pour the buffer in so that you have enough so it covers the gel completely. So I have my gel in there. I took the comb out so that I have actual little holes to load my sample into, and now I'm ready to load my samples. We're going to be using really small amounts of sample, and so we're going to use something called a micropipette. We know micro tells us that it is small, right? And so this is used for very small amounts of liquid. I have my pipette set to 35 microliters already, and that is the amount that I'm going to put into each of the wells. What I want to be careful of is when I put a tip onto my micropipette, that I use a different tip each time I get a new sample. I don't want any cross-contamination. So I'll use a separate one for each of my 5 samples that I'll be loading. So I'm going to go ahead and put a new tip onto my micropipette. So this is what it would look like when I'm getting ready to get my sample. The first sample I'm going to run is the red one. So you can see on your lab sheet it says lane 1 has the red sample, and that this is the standard DNA marker. So this is sometimes called a ladder, and we use it just as a standard. It proves to us that things are working in case all of our other samples don't run the way that we want them to. At least this tells us, well, something was working, so there must be a mistake with our other samples. So I'm going to go ahead and grab the 35 microliters out of this tube. And I have to be really careful when I load my samples because I need to make sure that I get it into the well, but I don't want to go too far because if I poke through the gel, it will mess up how the liquid DNA sample runs through the gel. If there's a hole, it's going to kind of leak out and we're not going to get the results that we need. So you have to be really careful. If you guys were here doing this with me, we would practice this a lot the day before because it is kind of complicated. So you guys will get a chance to see what this looks like once I get all of my samples loaded. But I've loaded the red sample into the first lane, and now I'm going to load the white one, which is the sample from the crime scene in this situation, into the second lane. So this lane is the one that is really important because we're going to be kind of measuring all of my suspect DNA against this crime scene DNA. And the idea is that if I can match up the crime scene DNA to the suspect DNA, then that would give me a good idea that that might be the person who did the crime. So my blue sample is suspect number 1. And I'm going to put this into lane 3. So you should have already read the scenario and have an idea of what the crime was that occurred, but we don't know who actually did this. And this is a technique that could be used in real life too, if there was various samples of DNA that could be compared to a crime scene DNA sample that was collected. Of course, they would probably use some other evidence as well prior to convicting somebody of something, but it definitely is helpful and something that is fairly new when we think about technology. I have 1 left, so I've got suspect 1 and 2 loaded. Now I'm going to have suspect 3, which is the yellow cap on my sample. All right, so now I have all of my samples loaded. So you can actually see when you're looking at it that in each of the wells, it looks like there is all of this kind of purple dye just sitting there. And once I actually turn on the electricity to this, the DNA, which is negatively charged, will start to move towards that red electrode, towards that positive electrode. And what's been done to these samples is they've been cut in specific places. And so you'll have some bands of DNA that are larger and some bands of DNA that are smaller. And the smaller bands of DNA should move quicker through that gel, and they should move further through that gel. And so what we're going to be looking for is a pattern where the suspect DNA looks identical to the crime scene DNA. And that would tell us that those two samples, when cut with these specific restriction enzymes, resulted in DNA pieces that were the same size and ran at the same speeds through my gel box. All right, let's keep going. The lights are back on now. So the last piece I need to do is I need to actually hook this up. And so I have a power source here, and I have a place to plug in both my black and my red electrode. So I'm going to plug in my black one first, and then I'm going to plug in my red. And I'm going to put the lid onto my box. And I want to make sure that I match the red up with the red that's on my box here and the black with the black. Definitely don't want to mix our positive and negative electrodes. Once I have this on, I'm just going to make sure everything looks like it's set, and I'm going to turn my power source on to 125 volts. And you may not be able to see this on the video, that's fine, I'll tell you what I'm seeing. On both ends here, I see kind of what looks like a metal wire. And if I look down to the side, I can actually see little bubbles coming off of them. And that indicates that my power source is working, right? There is electricity moving through this. And so if I were to let this run, I would let it run for probably about 35 or 40 minutes. I'm gonna actually see the purple bands start to move down the gel. And that tells me that my sample is moving down the gel. What I won't be able to see is all the other DNA bands that are moving at that same time. But that's what I'm gonna see when I put it on the UV light. That's the part that I'll actually get to look at. So I'll let this run for about 35 or 40 minutes. I'll take it out, dry it off a little bit, put it on the UV light box, and we'll get to see our results of our gel. All right, so now that we've let our gel run for about 35 or 40 minutes, I've taken it out, and so now I just have this gel outside of all of the plastic pieces that we started with. And what you should hopefully be able to see, but if you can't too well, I'll just tell you what I'm looking at, is it feels like a gel and I have purple bands at the bottom here. The most important thing when you're doing a gel electrophoresis is to make sure that those purple bands don't run off your gel, right? That they don't go away, because that means some of your sample may be gone, might have gone off of your gel. So I stopped it before it got too far, but I let it run a decent amount so that the bands that were different sizes could at least separate from each other. So now what I'm gonna do is I have this on a UV light box, and we're going to actually turn this on, and when we do that, we'll be able to see the different bands show up, and we'll be able to see our results. So we're gonna turn some of the lights off, and we'll show you what it looks like. Here we go. So what you should notice is you have the 5 different lanes, and lane 1 was our DNA standard marker. And for my purposes in this lab, I was really running that as a control just to make sure everything was actually working. You can use the standard markers for some more precise calculations in other labs, but for our purposes, we're just confirming that everything was working, and we can see there, we see lots of bands, so yes, things were working. Lane 2 is the one that is our crime scene, right? So that tells us this was the DNA that we found at the crime scene, which means that's the one I'm gonna compare to lanes 3, 4, and 5. And so your job is to look at lane 2, our crime scene, and compare it to lanes 3, 4, and 5, our 3 suspects, and figure out which of those 3 suspects you think committed the crime based on their DNA pattern there in the DNA bands. Once you've done that, you're gonna sketch out your gel, you're gonna answer the couple of analysis questions, and then we will come together as a class and talk about the conclusions and maybe what are some other things we could use DNA profiling for besides just solving a potential crime. Thanks.